Hi everyone, welcome to my channel Pure Biology, and today's topic is the types of immunoglobulin. Okay, and it is the topic related to immunology. And before this topic, I want to say that I have already uploaded many videos related to immunology in my channel, so please you check out that one. Okay. And here I also say thank you to all of you who have already subscribed my channel, and I want to say that the newcomers who are new in my channel, obviously share with your friends if this is helpful. Okay, so let's continue. The types of immunoglobulin. Here, before explaining, I want to say that the space is very short, so that's why I first explain the two types, and then after that I will explain another one. Okay. So here, the two immunoglobulins are, one is the IgG and another is IgM. IgG and IgM. So IgG, that is the most abundant immunoglobulin, most abundant. Generally 80% of this immunoglobulin is seen in the serum. Clear? So IgG is the maximum amount of immunoglobulin that is seen in the serum. Okay, and it consists of obviously two gamma chain, heavy chains, and two kappa and two lambda. Okay, two lambda light chains. So what is consisting of this? One IgG molecule, one molecule that consisting of two gamma heavy chains and either two kappa or two lambda light chains. Clear? Generally, four types of IgG molecules are seen. One is Ig1, IgG2, IgG3 and IgG4. So here, before explaining about this, one I am saying that this one is the less efficient, okay, but rather than three these are, okay. So this is the less efficient than these three. Okay, and how do we separate or how do we distinguish these four IgG molecules? By the presence of amino acid sequences. Okay, the presence of amino acid sequences that are generally present in the CH genes. CH genes. Okay, CH means about what is CH. This you can see in my previous video, that is related to the structure of immunoglobulin. You can see there. So CH genes, clear? The presence of, that means IgG1, they have one kind of amino acid sequence, and that is obviously absent in G2, G3 and G4. So, but that means the amino acid sequences are the actual substances by which we can easily distinguish these four IgG molecules. Clear? And the first three one, IgG1, G3 and G4, these have the capacity to cross the placenta and then protect the fetus. Okay, they have the capability to cross the placenta, or they are mobile, you can say. They have the capability to cross the placenta and protect the fetus. And IgG3, it has the capacity for enhancing the complement system, or you can say that this is a complement activator. Okay, this is a complement activator. And the IgG1 and G3, they can bind with the Fc region or Fc receptors. Fc receptors of the Fc region of phagocytic cells. Okay, then what can bind with the Fc receptors? The IgG1 and IgG3. So here the most important are IgG1, G3 and G4. G2 is less efficient. Clear? So this is the IgG molecule. Second one is the IgM molecule. Okay, it is seen generally very less amount, 5 to 10% in the serum, 5 to 10%, and this is also found where the surface of mature B cell. Okay, surface of mature B cell. First, it is seen generally 5 to 10% of amount this in the serum, but also it is seen the mature B cell, and here it acts with the IgD molecule. Okay, here it acts with the IgD molecules and serves an antigen specific B cell receptor. Okay, where it serves as an antigen specific B cell receptor, or you can say it acts as. Okay. Next, the monomeric, two kinds of IgM is seen. One is the monomeric, one molecule, and another is the pentameric. There are 5 molecules. So in case of monomeric, IgM is expressed as a membrane bound antibody on the B cells. Okay, these are seen in the B cell, and these are obviously monomeric. That means one single molecule is found in the B cell, or they remain as one single molecule. Okay, and generally, sorry for the disturbance. So, pentamer is a molecule, that molecule that is composed of such units. Such units means monomeric 5 IgM molecules that attach to each other to form the pentameric molecule. Okay, pentameric molecule, that means 5 monomeric, they attach with each other to form the pentameric molecule. Okay, and each consists of here 2 light chain and 2 heavy chains. Okay, and those are joined together by disulfide bonds. Okay, disulfide bonds, this is obviously additional disulfide bonds, and these bonds present between the Fc portion of 2 molecules. This is molecule, this is the molecules are just like this, 1 2 3 4 5. So Fc regions are connected. Okay, Fc regions are connected, and here the 2 Fc regions very close to each other, and that is very necessary for the complement activation. That's why IgM in the pentameric form is more active in the complement system rather than IgG. Clear? So this is the IgM. So IgM and IgD here we just learned here, and after that we will learn another 3 types. Okay, so continue with me. Okay, so there are the 3 types of immunoglobulin. One is IgA, another is IgD, and the last one is the IgE. So IgA generally present 10 to 15% in the serum. Okay, 10 to 15% in the serum, and it is the predominant class of Ig that is secreted or that is seen in the external secretion like saliva, tears, mucus, breast milk, etc. Okay, so IgA is one kind of immunoglobulin that are secreted or that are seen in the external secretion. Okay, external secretion. There are 2 types of IgA. One is IgA1, another is IgA2. Okay, and in the serum we found in the form of monomeric molecule. Okay, or individual molecule. But in the external secretion they remain either dimeric form or the tetrameric form. That means they remain either as the dimer or tetramer. And here obviously just like IgM, there are J chains also present by which those are connected. Dimer means 2 molecules that are connected, and tetra means 4 molecules are connected. Okay, and these are seen in the external secretion, And in the serum, what type of IgA we see? Generally in the monomeric form. Clear? Now, this secreted IgA, this secreted or that are found in the external secretion, this secreted IgA generally transported through the epithelial cell into the intestinal lumen. Where they are transported? To the intestinal lumen and through the epithelial cells. And here one Fc receptor is very important that help in the transportation. Okay, and this secreted dimeric IgA that containing one not only one, the J chain and Ig receptor. Okay, so in the dimeric form we can see the J chains and the Ig receptor, and these are found in the mucosal epithelial cells. They are bind to each other. Simply, just like IgM, two dimers of IgA that are connected by the obviously J chain. Okay, so J chain is found there obviously, and this endocytosis. Okay, endocytosed here. Here this endocytosed and transported in the vesicles to the luminal surface, and the poly Ig receptor is cleaved and cytoplasmic domains are attached to the epithelial cells. So what is happening here? When from there actually these are the found where here the secreted the IgA molecule that transported through the epithelial cell into the intestinal lumen, and by what? By a specific receptor, that is the Fc receptor. Okay, this Fc receptor on the mucosal epithelial cells actually helps in this process. Okay, and here the J chain that is found or that is seen in the dimeric form of the IgA. And what happening there? Their endocytosis is seen. Endocytosis process is seen. And after that, these are transported in the vesicles, and after that to the luminal surface, where the poly Ig receptor, the receptor generally cleaved, the receptor that carry the IgA molecules from one place to another place, and after that the total completion of the function, this receptor, poly Ig receptor, is cleaved. Okay, so what remains there? Only the domains, cytoplasmic domains, these are remaining there. Okay, and this domain which carries the IgA molecule that released into the lumen. So domains, with the domains the IgA molecule present, and this released in the lumen. Okay, so what is seen here? After transporting this poly Ig receptor that is totally cleaved, but cytoplasmic domains are there, and with the domains the IgA molecule that are bind there, and these IgA molecules just released from the domain into the lumen. Clear? And in that way the secreted IgA molecules activate the enzymatic reactions, or it helps in the production of the antibody molecules. Okay, so these are the actual function, or how they functions, IgA molecules in the external secretion. This is the process. After transportation, the IgA molecules release from the domain, and then it activates in the formation of the antibody molecules. Okay, next, IgD molecules. These IgD molecules actually 0.2% is there, where in the serum. Okay, so IgD molecules actually functions like IgM or with IgM obviously, and these are the major membrane bound IgD molecules, major membrane bound immunoglobulins. Okay, sorry. So here the IgD molecules, these are the actually together with IgM molecules, this expressed by the B cells, and these are the major membrane bound Ig molecules. Okay, and they functions with the IgM. Next, IgE molecule. It immediates the hypersensitivity reactions. Okay, that means its function is IgE molecule. Its function is only to produce the hypersensitivity reaction just after the antigen entering into the body. Okay, so IgE generally binds to Fc receptor. Okay, on the membrane of blood basophils and the tissue mast cells, and cross linkage of the receptor bound IgE molecules by antigen induces the degranulation of the cells. What is seen here? That it generally IgE generally binds with the Fc receptor. Okay, sorry for the disturbance. So actually here IgE that binds with the Fc receptor, and where it binds generally the two kinds of cells, blood basophils and the mast cells. Its membranes there are Fc receptors, and this IgE that binds with the Fc receptor. Okay, and the cross linkage of the receptor bound IgE molecules by antigen induces the degranulation of the cells. Okay, degranulations of the cells, and this results the pharmacologically active mediators, giving rise to allergic manifestations. That means when the Fc receptor and IgE molecules both bind on the membrane of the blood basophils and the tissue mast cells, what happening? These cross linkages create the degranulations of the cells. Okay, or you can say the binding of this creates the degranulations of the cells. Which cells? The basophils and the mast cells. And as a result, what happening? The pharmacologically active mediators or symptoms, you can say the symptoms or these hypersensitive reactions are seen. Okay, hypersensitive reactions are seen, and so that's why the allergic manifestation is occurring. Okay, allergic manifestation means the allergic or allergic symptoms, you can say, these are found in the body. So this is the IgE molecule. Okay, and IgD is just like same IgM. It functions with IgM, and IgA, it functions in two ways. One that present in the serum in the monomeric form, it is directly functions, and what another form, these are the dimeric or the tetrameric form that present in the external secretion. They form in the following way: that first bind with the Fc receptor. Where is that? This is the Fc receptor. With the help of J chains, they bind to each other, and then it goes to the lumen. Okay, luminal surface of the from vesicles to the luminal surface, and here the poly Ig receptor that totally cleaved, and the domains are only present there, and the domains containing the IgA that released. Okay, or IgA released from the domain. Okay, there in the lumen. Okay, so this is then enzymatic activity is seen. So these are the actual types of the immunoglobulin. Totally I have explained here. I think you understood it properly, and if there is any problem is created, you can consult any immunology book. Okay, so I will explain another topic related to immunology in my next video. So continue with me.